So let's talk about this. TEM on the dried sample. So that's a cellular level cytotoxicity study. So I have a big review. It's a chemical society review on nanotoxicity, how you systematically study the toxicity of nanoparticles. Okay? Then what is the next step? You go to the animal studies. And in animal studies also, before you do the therapy so on, you want to make sure that the bare nanoparticle is not toxic. Because cell study is not enough. Cell toxic only at cell level. What about if these nanoparticles react with extracellular matrix that bind the cells together and damage that? Okay? Or collagen. So you want to do it at animal level. So animal level, you inject the nanoparticles, the bare one, to see how the nano formulation itself is, or the one where the therapy you can activate from outside. That means without activating the therapy, you use it, controlled, and you want to see if the animal's behavior first has changed. If it is toxic, the animal may not eat, may be hungry, not hungry, may be acting weird, somehow, may be very lethargic, maybe not sleeping, so all these behavior changes. First we look at behavior changes, right? And then if the behavior change shows there is no behavior change, that means it's not toxic, right? But still, you don't know if there are any kind of, it's a behavior, right? But you don't know, maybe it has caused damage to some organs, and you don't know, that may not have affected the brain somehow. So you sacrifice the animal, and you look at different organs, specimen of the liver, kidney, spleen, heart, all these tissues you look at under a microscope and see if you see any damage evidence of a structure. Okay? And again, if you want to control, you can look one animal without nanoparticles, one with nanoparticle, compare the two after same days, right? And from there you can see there's nothing coming out in urine, nothing coming out in stool, right? So you examine that as well. And then you do pathological examination, sacrifice, and look at it. So this is the way you kind of progress in any nano formulation of drug. It's a learning process. And you need to be able to do all yourself. This is where, maybe you can make materials, nano formulation, characterize, do DLS and all these others. And then you work with a biology friend who can do these cellular studies or animal studies. As some of you said, well, I don't know, I don't want to touch mice, And as I said, you got to wear gloves because sometimes they bite you if you're trying to inject or clamp something. So you got to be careful, So my students, if they're working in animal, in the US there are strict regulations even working with animals. You need to take a course and you need to kind of be certified to handle the animal and do animal experiment. Same thing, our storage area, we have to have a license to be able to store the animal and do the studies. So eventually then, just for your information's sake, where do we go? Suppose you have done testing in mice and everything looks great. Where do you go from there? Well, you would go to a bigger animal, first step, and then prove that it works in bigger animal, then you have all these data, and so then they will grant you permission to try on humans. That's what is called trial. And the human, there are volunteers. And the volunteers generally are people who have given up on other treatments, and they say, what have I got to lose? Let's try it. And sometimes there are people, they can be paid for the test. So they sign consent that, okay, I give permission to be tested, and in lieu of that they get some money. Okay, so they are ready to be tested, and then you conduct trials, look at toxicity, the same repeat experiments that you did with animals. You don't sacrifice the human, you just do biopsy. You take a little piece and study that, right? So, and then with all the results you submit, and you get approval to actually treat patients by that. So it's a long process. So first when you come up with the idea of what nanoparticle you make, it is easier if you are providing nanoparticles as a carrier. You're just loading it and delivering it. Then it's somewhat a different procedure. For approval, anything which is external, and you inject it in either directly into the tumor or systemic, and this is a dispersant which could be nanoformulation or just mixed with a surfactant to disperse the carrier. So we're talking about nanoformulation maybe, so they go around every place, but even without targeting, this is what is intrinsic targeting in this case. These porphyrins have higher affinity for tumor cells, cancer cells. So they may go around, but if you wait for, let's say, a day or so, then every place has cleared out, and these are sitting still in the cancer cell. Then at that point, you irradiate with light, and it does this thing, right? And then the cancer cells break down. The tumor is shrinking. Then these photosensitizers come out of the cancer cells, and they're floating around other places. So after you have treated, you want to wait for it offer. One of them is that you can localize with imaging, these dyes up, and you can also use upconversion photodynamic therapy. That means, like our nanotransformer together with photosensitizer, you only need half energy, longer wavelength. It goes to excited state by 2 photons, emits the light in the blue in the region. A blue light can be absorbed by the photosensitizer. So it's upconversion photodynamic therapy we can do. We can build image, can put targeting to enhance more accumulation in the tumor, and so we can favorable effect.